Stability testing and regulatory compliance in the pharmaceutical industry is a very vast topic, and it is very challenging topic. It is very much required that to prove the safety and efficacy of the pharmaceuticals, the stability testing and regulatory compliance is required. Welcome dear viewers to this video, and in this video we are going to learn the key points or key understandings for stability testing for pharmaceuticals and regulatory compliance. So let's start with the video with some basics. Stability testing involves the testing of pharmaceuticals by storing at different temperatures and humidity conditions to study its effect onto quality, stability and efficacy. Stability of the pharmaceuticals means ability of the drug or drug product to maintain its physical, chemical, therapeutic and microbiological properties during shelf life. So the product should be the same quality at the time of manufacturing and till the end of shelf life, so that the drug product can be safe and effective during this shelf life. Then accelerated testing failures. So as I mentioned, this video is for the key points to understand about stability testing and regulatory compliance. So it will be a short video and key points will be discussed. Accelerated testing failure. So if the product fails at accelerated condition, it may require intermediate stability testing for verification. Intermediate stability condition samples are loaded along with the accelerated and long-term testing. Then NDA ANDA submission. It requires 6 months data of accelerated and real-time stability studies, and further the intermediate data may be required. So it is not only for NDA ANDA, it is generally considered for all the markets. Intermediate testing. So intermediate testing is done if the accelerated conditions show significant failures. Excursion tolerances. What is the excursion tolerance for the stability chamber variations? This may be because of the background of the stability chamber. So 24-hour tolerance for stability chamber variations is allowed. What is significant change? So significant changes are those changes which occur because of the stability conditions and which may be related to the physical, chemical or microbiological properties of the drug product during the stability studies. For example, decrease in the potency or assay, increase in the impurity levels, or failure to meet the dissolution specification, failure to meet the or failure to maintain the description or appearance. These all come in the significant changes. So accelerated testing changes may require intermediate studies. Real-time changes may risk on approval. So if the product is not stable or if significant changes occur at accelerated testing conditions, then you can prove or you can justify the changes because of the accelerated condition based on the intermediate data. But if the significant changes occur at real time condition, then there will be a risk for the product approval. Then 5 degrees Celsius refrigeration. It is not a special case for stability testing. Water loss considered in stability testing for aqueous based products in semi-permeable containers. So whenever you are working onto the products which are stored in semi-permeable containers, that time water loss will be considered for stability testing. Key regulatory requirements: bracketing and matrixing. It can be used for packaging configurations. Bracketing and matrixing is a way to reduce the stability samples and to prove that the in between packs or in between batches samples are meeting the specification if the extreme conditions are meeting or some matrix samples are meeting the requirements. Shelf life calculation. So shelf life is calculated based on ICH-Q1E principles using 6 months of accelerated data and 12 months of intermediate data. Accelerated data for 24 weeks. 6 months of accelerated data can support 24 weeks of stability data. It is very important to understand that how much data for accelerated stability testing can support the shelf life. So 6 months of accelerated data can be used to support 24 weeks of stability data. Then API batches. 2 API batches are required for manufacturing 3 batches of product for each strength of the drug. For example, if you are working on a capsule formulation in 3 different strengths like 100 mg, 200 mg and 300 mg, then for making all the pivotal batches or exhibit batches, 2 API lots or 2 API batches will be required. Key points on drug product manufacturing. Pilot scale batch storage. So this you can say it as bioequivalence batch. So samples should be stored for 1 year after approval or 5 years for bioequivalence studies. Using different API sources. Typically, there is no need for using the different API sources, but if the 3 batches are required to be made with different packaging materials, then there is typically no need for different packaging unless it affects product performance. But the API batches should be 2. The main understanding out of this point is if packaging material is affecting the drug product quality, then packaging material lots are also required to be used like we are using the 2 different API sources. Stability for multiple API sources. So if you are using the alternative APIs, that time stability data for 3 batches needed for each API source. Also, it is generally regarded as uh, making the 3 batches on primary API source and 1 batch of the B strength for the other API source. But many of the regulatory bodies or regulatory agencies ask 3 batches. Scale up batches in non-sterile facility. So it cannot be possible that scale up batches are done in non-sterile facility and exhibit batches are being done on sterile facility. So sterile product must be manufactured in sterile facilities to maintain the sterility. Intermediate material consistency. To prove the intermediate material consistency, separate intermediate batches required for each strength of the product. The example is like tablet formulation or capsule formulation coming in different strengths. So need to make the separate intermediate batches to make those common blend or make those strengths using the same blend. For example, 2 strengths are there, 100 mg and 200 mg, and both are made from the same blend. So it is not like that only one batch will be made for complete common blend and different batches will be compressed. So it is not like that. Always you have to go for separate intermediate batches or separate lubricated blend for making each strength of the product. Then preservative effectiveness and extractable testing. Primary batches tested for preservative effectiveness and included in stability studies. CGMP batches for API retested Three primary CGMP batches required to support the API retest stability. So it is very important to know that three primary batches will be required to support API retest stability. These were the common points for stability testing for pharmaceuticals and regulatory compliance. Many times you may come across the questions on these points, and always remember the key points for regulatory compliance. This was a video regarding the key points only. I have made many other videos and those are available on the channel. So I request you to go through those videos so that you will have complete understanding on stability testing. Thank you for watching the video.